Thank you for the introduction. Just to give you an overview over a few numbers, currently it's estimated that around 55 million people are affected by Alzheimer and other forms of dementia. And with an aging population, this number is going to increase exponentially over the next coming years. Also with an increasing number, it's likely that the death rate from Alzheimer's is also going to increase. So here's a graph from 2019 where the death rates are depicted and the numbers are showing the people of the number of deaths per 100,000 people. So this shows us that we want to look at preventative interventions using sensitive biomarkers to face this increase in individuals with dementia. So dementia can be subdivided into different severities. So we have mild, moderate, and severe Alzheimer disease, and during the disease progression, we see shrinkage in the cerebral cortex and the ventricles are getting enlarged. And it has also been shown that individuals with Alzheimer's disease have increased tau tangles and an accumulation of amyloid beta. So Alzheimer has multiple risk factors, but it's estimated that around 70% of the overall risk is due to genetics. And the strongest genetic risk factor is ApoE4. There are other forms such as ApoE3, which is associated with low risk, and ApoE2, which is known to be preventative. Before structural changes occur, we have seen that impairment in the cerebral metabolism and vascular function are visible. So here we have a nice graph where we see here the structural changes on this line, and we see here the vascular changes on the orange line, and metabolism is here on the darker blue line. So if we now find a time point where we have where we see early onset changes, we can start with the preventative interventions. So in Alzheimer's disease, it has been found that there is a decrease in CBF compared to healthy controls in some regions, and for oxygen extraction factor, which we have heard a lot about in the last few days, there has been found a decrease in the PET, positron emission tomography, in some regions in Alzheimer's disease healthy controls. For OEF, there has been also another study which used trust to show also the same decrease. So compared to normal during the disease progression from normal to dement individuals, there has been a decrease in the oxygen extraction fraction. So how does now these two biomarkers look in individuals who are at higher risk of developing Alzheimer's? So in individuals who have the ApoE3 allele, shown that there was regional hyperperfusion in McKiernan has shown that. And on the right side, we have a which looked not only at individuals with ApoE4, but they subdivided it further into the amount of ApoE4 alleles the individuals had. And the higher the risk factors, so the more ApoE4 alleles they had, the lower was their OAF value. And I've mentioned already two different methods of how we can calculate oxygen extraction fraction. There is another one commonly used, and that's with QSM, quantitative susceptibility maps. So I just listed here some disadvantages and advantages of the three used methods. There are other methods, of course, as well, as we have heard earlier today. So for PET, that's still the gold standard. However, it requires an active tracer. For OEF, it requires labeled oxygen, which has a half-life time of only 2 minutes, which requires basically a cyclotron on site to measure OEF. TRUST is non-invasive. However, it has no spatial resolution. So we look at one single value in the sagittal sinus, and we get only a global OEF estimation from that. QSM can give us regional values inside veins, and it only requires a standard sequence on the scan. It has a complex image reconstruction, which we have to do beforehand. So how can we get oxygen extraction fraction from QSM? So we use the paramagnetic properties of deoxyhemoglobin, and basically the deoxyhemoglobin OEF is proportionally correlated to the susceptibility values inside veins. So we measure then the susceptibility shift between veins and water to estimate oxygen extraction fraction. So we use this equation here. We can read out from the QSM image the susceptibility value inside veins and subtract it from the susceptibility values inside water, which we can get from the ventricles. We have a constant, which is the susceptibility difference between fully oxygenated blood and fully deoxygenated blood. We need a hematocrit, and then we can solve this equation for oxygen extraction. And for the QSM reconstruction itself, we have heard a little bit earlier about QSM this morning. We need a gradient equal sequence where we save the phase and the magnitude separately. We do then phase unwrapping and a background field removal to get to the local field map, and then we have to do a dipole inversion to get to the susceptibility values. As soon as we have the QSM image, we can run Vascular, which looks for bright and tubular structures to segment veins inside QSM. And then we can use these the map to estimate oxygen extraction fraction inside the veins. We used here the data of 141 actually participants from the Prevent AD dataset. We used the 2019-2020 version. At the initial scan in 2011, the participants had to be 55 years or older, had to have a MoCA score of 26 out of 30, and they had a family history of Alzheimer, where either their parents or siblings were diagnosed with Alzheimer. They used a 3 Tesla Siemens scanner, and we used the gradient echo field maps to estimate QSM, and a P-Castle sequence to calculate. So here are some demographics of our data. We used 141 participants. We subdivided that into individuals who had ApoE 4 and ApoE 4.3 and compared them to individuals who had ApoE 3.3. For our results, we found that individuals with the ApoE 4 allele had significant lower OEF in the frontal, occipital, parietal, and temporal lobe, and also in a 6 millimeter distance around the hippocampus. For our CBF results, we have not found any significant changes between our two groups. However, we have a slight increased CBF values in the frontal, parietal, and temporal, but was not significant. We further were interested if we also see a correlation between our OEF and CBF values to the delayed memory of these individuals, which was measured with the argon battery. And we have not seen any significant results. However, there was in the occipital, temporal, and in the area around the hippocampus for individuals with ApoE3, where they seem to have higher OEF values for lower delayed memory scores. If we look at the CBF, we also have not seen any significant results. Our region of interests, again, the ApoE3 group has shown a tendency of having slightly higher CBF for individuals with lower delayed memory scores. So to summarize the results, we have seen lower OEF in the ApoE4 group, which indicates that there is an imbalance between the blood supply and the oxidative metabolism. However, we have not found any significant findings in CBF, and it's possible that we had a subgroup which kind of experienced hypoperfusion, while others already have initiated the decline phase. And also we had fairly large regions where we looked at the CBF. So it might be if we decrease the size of the region that we actually see significant results. And we have not seen any significant findings with cognition. However, these individuals, most of them were still cognitive normal. So we had 14 MCI converters in the data set, so these ones might not have been our results here. We had some limitations. We had an unequal sex distribution. We had twice as many females as males. We had a small-ish sample size. So when we divided our group into ApoE4 and ApoE3, we could not further subdivide it into males and females. However, there is currently a new release of that data set, so we can start looking longitudinally at the oxygen extraction fraction changes and also at the CBF changes. There are now more MCI converters, so it would be also interesting to subdivide it by MCI converters versus non-MCI converters, and maybe combine it with other biomarkers as well. And in conclusion, we have seen that OEF might have a greater sensitivity to detect the effects of the ApoE4 genotype compared to other biomarkers such as CBF. Thank you all.